Hello dear students, I am Dr. Mohammad Ismail, Department of Zoology, J.A.T. Arts, Science and Commerce College, Malegaon. Today in this video, we are going to see some MCQs on enzymes, which is the chapter of biological chemistry, TYBSc Zoology. Question 1. What EC stands in nomenclature of enzymes? Options are A. Enzyme concentration. Option B. Enzyme commission. Option C. Enzyme catalyst. And option D. None of the above. So the correct question is option B. Enzyme commission. EC stands for enzyme commission in nomenclature of enzymes. We have seen that classification of enzyme EC1, EC2 up to EC6. So enzyme commission is the correct answer. Question number 2. EC1 denotes which class of enzyme? Options are option A transferase, option B isomerase, option C hydrolase, and option D oxidoreductase. So the correct answer of this question is option D oxidoreductase. Look, we have seen 6 main classes of enzyme and they were denoted as EC1 to EC6, and in this question we have seen EC denotes oxidoreductase. Question number 3. Dash enzymes are also called as synthetase. Options are option A lyases, option B hydrolases, option C ligases, and option D oxidoreductase. So which one of these called as synthetase? So the correct answer of this question is option C ligases. Ligases are enzymes, for example DNA ligase, which in DNA replication joins two Okazaki fragments of DNA molecule and make it a single linear chain or polymer of DNA nucleotides. So this is also called as synthetase. Question number 4. Allosteric enzyme possesses two binding sites, whether it is true or false. So the correct answer of this question is option A true. This type of enzyme possesses two binding sites. The active site where substrate of the enzyme binds and the allosteric site where the effector or modulator of molecule binds. So this type of enzymes is known as allosteric enzymes. Next question, question number 5. Which of the following is not the property of allosteric enzyme? Options are, these are some properties of enzymes and we have to find out which property is not belongs to allosteric enzyme. Options are option A, it has two binding sites, active site as well as allosteric site. Option B, they are larger in size and mass also. Option C, conformational changes occur in active site after the attachment of modulator. And option D, they show hyperbolic curve when we prepare the graph between concentration of substrate and enzyme activity. So it shows hyperbolic curve. So of which of the following is not the property of allosteric enzyme? So the correct answer of this question is option D. Basically, allosteric enzymes are shown on a graph as velocity against substrate concentration, they show a sigmoid curve rather than usual hyperparabolic curve. Allosteric regulation of enzyme is crucial for the control of cellular metabolism, and not all enzyme possesses sites for allosteric binding. Those that do are called as allosteric enzyme. Means all enzymes are not allosteric enzymes, but of these few enzymes are allosteric enzymes which having two binding sites. One is active site, another one is allosteric site. Allosteric enzymes are generally larger in mass than other enzymes and typically comprise multiple protein subunit. They are having big size and big mass also. Based on modulators, they can be classified into two groups: homotropic as well as heterotropic allosteric enzymes. So the correct answer of this question is D. They show hyperparabolic curve, which is not the property of allosteric enzyme. Now next question is question number 6. The substrate and modulators are different molecules in dash dash enzyme modulation. The substrate and modulators are different molecules in dash dash enzyme modulation. Options are homotropic enzyme modulation, option B heterotropic enzyme modulation, option C positive allosteric enzyme modulation, and option D negative allosteric enzyme modulation. So the correct answer of this question is option B heterotropic enzyme modulation. Look, the name itself indicating that heterotropic means there are uh, different molecules. Substrate is different molecule and modulator is different molecule. So in heterotropic enzyme modulation, these two compounds are different one. Next one, in positive enzyme modulation, after binding of the modulator, affinity of the enzymes to substrate dash dash dash. Options are increases, option B decreases, option C remains constant, and option D none of the above. In positive enzyme modulation, after binding of the modulator to the enzyme, affinity of the enzyme to its substrate increases or decreases or remain constant. So the correct answer of this question is option A, increases. Allosteric modulation is a phenomenon displayed by enzymes that have multiple binding sites. As we have seen that allosteric enzymes are having two important sites. One is for attachment of substrate, another is for modulator. There are two types of modulations, positive as well as negative. When affinity of the binding site for a substrate is increased, then it is positive modulation. When it is decreased, then it is called as negative modulation. An example of positive allosteric enzyme modulation is the binding of oxygen to hemoglobin molecule. Hemoglobin is a protein in RBCs. When one oxygen molecule bind to the ferrous ion of heme molecule in each of the four chains of a hemoglobin molecule, then it increases the affinity of oxygen, and affinity of hemoglobin to the oxygen is increases. So it is the best example of positive enzyme modulation. Deoxyhemoglobin has a relatively low affinity for oxygen, but when one molecule binds to a single heme, the oxygen affinity increases, allowing the second molecule to bind more easily and the third and fourth even more easily. The oxygen affinity of 3-oxyhemoglobin is 300 times greater than that of deoxyhemoglobin. By the same process, the ability for hemoglobin to lose oxygen is also increases as fewer oxygen molecules are bound. So the correct answer of this question is option A. Next one, question number 8. Binding of oxygen to hemoglobin is an example of dash enzyme modulation. Options are heterotropic positive enzyme modulation. Option B, homotropic negative enzyme modulation. Option C, heterotropic negative enzyme modulation. And option D, homotropic positive modulation. So the correct answer of this question is option D, homotropic positive enzyme modulation. The first thing which we have to clear is that what is the difference between homotropic as well as heterotropic enzyme modulation. In allosteric enzyme modulation, if the modulator is same as that of substrate molecules, means substrate itself act as a modulator, so this type of enzyme modulation is called as homotropic enzyme modulation. And if substrate and modulator molecules are different one, so this type of enzyme modulations are called as heterotropic. But look, the question is binding of oxygen to hemoglobin is an example of dash enzyme modulation, which just one question before we have seen that oxygen itself acts as a modulator and it is a substrate also. So it increases the affinity of second oxygen molecule to heme. So this type of modulation is known as homotropic positive modulation. So the option D is the correct answer. Next one, question number 9. The enzyme is inhibited by the end product of the reaction and the reaction is called as dash inhibition. This type of reaction is called as dash. Options are option A, negative allosteric inhibition. Option B, positive allosteric inhibition. Option C, feedback inhibition. And option D, all of the above. The enzyme is inhibited by the end product of the reaction. So this reaction is called as, the correct answer of this question is feedback inhibition. Option C is the correct answer. In some multiple enzyme systems, the enzyme is inhibited by the end product whenever its concentration is above the requirement of the cell. So the velocity of the reaction can be controlled by the amount of product that is needed by the cell. The lower the requirement is, the slower the reaction goes. When the regulatory enzyme reaction is slowed, all subsequent enzymes operate at reduced rates as their substrates are also depleted. Rate of production of the pathway's end product is thereby brought into balance with the cell's need and is called feedback inhibition. For example, biosynthesis of L-isoleucine from L-threonine in bacterial cell. So the correct answer of this question is option C, feedback inhibition. And the last question, question number 10, which among the following is not the example of covalently modulated enzyme? We have seen different types of enzyme regulation. So this one is also the type of enzyme regulation, covalently modulated enzyme. So which one among the following is not the example of covalently modulated enzymes? Options are option A, phosphorylation, option B, adenylation, option C, ADP ribosylation, and option D, oxidation. So the correct answer of this question is option D, oxidation. Look, oxidation is a reaction which is basically carried out by oxidoreductase enzymes, and the remaining three options are the examples of or the type of covalently modulated enzymes. Phosphorylation example, kinase enzyme basically involve. Look, in this type of modulation, covalently modulated enzymes, in this type of modulation, a second enzyme is also needed for the activity of main enzyme. Phosphorylation, adenylation, and ADP ribosylation, like and methylation. These are the examples of covalently modulated enzyme. In this, the second enzyme basically make changes or some addition or deletion takes place in the main enzyme, and the enzyme reaction is carried out. So this type of enzyme regulation is basically carried out with the attachment of some molecules like phosphate and adenyl monophosphate, ADP ribosylation is also, methylation also takes place. So this type of enzyme modulation is known as covalently modulated enzymes. So the correct answer of this question is oxidation. Oxidation is a reaction which is carried out by oxidoreductase enzyme, and it is not the example of covalently modulated enzyme. So thank you all of you for watching this video, and best luck for your examination. Thank you all.